Welcome back to Prokaryotes, Part 2. We're going to be talking about the cell envelope, the cell wall, and the cell membrane. The cell envelope is everything that divides the inside of the cell from the outside of the cell, and that is composed of a cell membrane. All cells have a cell membrane, and potentially a cell wall. When we talk about the cell envelope of bacteria, most bacteria are divided into two groups, gram-positive and gram-negative, and that's based on Graham's stain. Graham's stain is a technique used to differentiate these bacteria, and it was perfected by Hans Christian Graham. He was the one who discovered this stain, and so he is also credited every time we write it. So when you do say Graham stain, the G in Graham stain should be capitalized. The Graham stain, whether your organism is gram-positive or gram-negative, is going to stain differently. The process is add crystal violet. Crystal violet is a positive stain, so it's going to stain the cell envelope of both gram-positive and gram-negative cells at this point. Then we're going to add iodine. Iodine is the mordant. You can consider the mordant as mortar, like mortar of bricks, because it cements the crystal violet into the peptidoglycan of gram-positive cells. Peptidoglycan is this thick outer layer here, and so that is the gram-positive with the thick peptidoglycan. Now, gram-negative cells have a very thin layer of peptidoglycan, and so there is some cementing there, but not nearly as much. Not nearly as much peptidoglycan, so the crystal violet doesn't stick in as much. This was the important step that Graham discovered with his Graham stain technique. And then when we decolorize, when we wash with alcohol, it will remove the crystal violet from the cells. And because the gram-negative cells have an outer membrane composed of phospholipids, a phospholipid bilayer, that's going to be solvated by the alcohol, and so the crystal violet is going to be stripped much more easily. Whereas because the crystal violet was cemented in the thick peptidoglycan layer of the gram-positive cells, it does not decolorize quite as quickly. So at this step then, the cells are two different colors. One is colorless, the gram-negative, and one is purple, and this is how we differentiate the two cells. And we could view them at this point, but if we did, we would not be able to see the gram-negative cells because we have stripped all the stain back out of them. And so we use a counter stain, in this case safranin, because it's pink and it's a weaker stain, and the safranin will stain the gram-negative cells. It will stain the gram-positive too, but because it's pink, it won't overpower the purple, and so we end up with two different color cells. We end up with pink for gram-negative and purple for gram-positive. And I always say, I remember these because of the 3 P's. Gram-positive is purple, because it has a thick layer of peptidoglycan. And here's the stains again with more color, and here you can see the gram-positive has a thick layer of peptidoglycan, whereas the gram-negatives have a very thin layer of peptidoglycan, and instead they have an outer membrane composed of phospholipids. And if you did your Graham stain properly, you should be able to see both gram-positive and gram-negative cell types in a mixed culture. This is more like what your Graham stain control will look like when you perform this technique in lab. We have gram-negative Escherichia coli bacilli and gram-positive staphylococci. And notice that true staphylococci arrangement, childlike, grape-like clusters, staphylo over here, staphylo arrangement. So, is this organism gram-positive or gram-negative? Gram-positive or gram-negative? Yes, it's gram-positive. Positive purple peptidoglycan. Is this organism gram-positive or gram-negative? Well, we see two different colors there, but remember, only peptidoglycan is going to stain purple. And so if this is a clinical stain, and it is, this is a human sputum stain, so we have some epithelial cells there and some mucus and other gross stuff, and lots of gram-positive bacteria. And the pink, then, because it's eukaryotic cells, it's human cells, does not have peptidoglycan, it will not stain pink. So the purple is gram-positive purple peptidoglycan. It's a bacterium. Now here we have those same neutrophils that we saw earlier in part 1 and gram-negative diplococci. So again, the eukaryotic cells do not have peptidoglycan, so they will not stain purple, but in this case the bacteria also do not have a thick peptidoglycan layer, so they do not stain purple either. This one's a little iffy. A lot of times we require students to use a gram stain control on their slides so that you could decide if this was more pink than purple. When you stained it, you would compare it to your control. I would think that this would be a gram-negative bacterium. And so if I said it was gram-negative, does that mean that it has peptidoglycan or no peptidoglycan? No peptidoglycan. Remember, the gram-negative cells have a very little amount of peptidoglycan, and sometimes as microbiologists we get lazy and we say it has none. So positive purple peptidoglycan, negative no peptidoglycan. Okay, so I keep talking about this peptidoglycan, but what does that actually mean? Well, it's in the name. When you talk about glycans, we're talking about long chains of sugars, and they're repeating sugars. You don't need to know N-acetylglucosamine and N-acetylmuramic acid. That's what's abbreviated there. But you do need to know that it's long chains of sugars. That's the glycan. And when you have long ribbons of sugars, that doesn't create a cell wall. That just is long chains. So they have to be cross-linked to make this thick mesh network. And this thick mesh network is cross-linked using short chains of amino acid called peptides. They're not long enough to be proteins, they're just peptides. So that's the peptido and peptidoglycan. And what is the purpose of this? This is a true cell wall. It is going to prevent the cells from imploding or exploding, and a lot of times when we say lysis, we're talking about exploding. Exploding, lysis mean the same thing. So while gram-negatives or gram-positive cells have a thick layer of peptidoglycan, gram-negative cells have a thin layer of peptidoglycan, so they need something else to prevent them from lysing. So outside that peptidoglycan layer, they have another layer. It's called an outer membrane. If it's a membrane, it's composed of phospholipids, and that is a phospholipid bilayer creating an outer membrane. Phospholipids have a hydrophilic head and a hydrophobic side chain, and that means that only water and uncharged molecules are going to make their way through that without regulation. And so that outer membrane of a gram-negative cell is selectively permeable. There are porins inserted there that allow the cell to regulate what comes and goes if it is a charged molecule or a large molecule, but only water and uncharged molecules will pass through there, and so it is selectively permeable. Now also in these two cell envelopes are things sticking out of them. Notice on the gram-positive side we have teichoic acid and lipoteichoic acid. Those are, even though they say acid, those are sugars. They're acidic sugars. They're charged sugars sticking out of there. And those have significance. On the flip side then, the lipopolysaccharides that are sticking out of the phospholipid bilayer of the outer membrane of the gram-negative cells, lipopolysaccharides are lipids stuck down in the phospholipid part of the phospholipid, and they have long chain sugars that hang out. That's the polysaccharides. And these are things hanging out of the cell envelopes, and they each have a meaning to the cell, which I'll circle back to, which is part of why we do a gram stain. It doesn't just tell us if it has peptidoglycan or no peptidoglycan. It also tells us about the entire cell envelope. So speaking of peptidoglycan, when we're talking about bacteria, gram-positive and gram-negative, versus eukaryotes, peptidoglycan is only found in bacteria. So you really could say the 4 Ps, positive purple peptidoglycan prokaryotes, although it's only found in bacteria, which messes up my alliteration. Positive purple peptidoglycan prokaryotes. And the significance of that is then that if we could treat a person with a gram-positive infection with a drug that would target peptidoglycan. Because if only bacteria have peptidoglycan, then that means we could give somebody a drug, it would harm the bacterium, and it would not harm us. Also, because peptidoglycan is only found in bacterial cells, that also means that the body would recognize that as being foreign material, and so gram-positive infections don't often go unnoticed by the immune system. Also, in that cell envelope, between the cell membrane, now remember all cells have a cell membrane, it's composed of phospholipids, and the peptidoglycan layer is the periplasmic space. Now the periplasmic space is another one that it's large in gram-negative cells. Remember, that's where the periplasmic flagella are in gram-negative spirochetes. But it's also one of those things that is so narrow in gram-positive bacteria that we often get lazy and say that gram-positive bacteria do not have a periplasmic space. They do, it's just so small that we say that they don't have one because we're lazy. So the whole thing is an envelope. The gram-positive cells have a cell wall composed of peptidoglycan. The gram-negative cells have instead only thin peptidoglycan and an outer membrane. And then they each have things attached. So we can summarize that. Again, gram-positives have thick peptidoglycan layer with teichoic and lipoteichoic acid stuck in, and a thin to no periplasmic space, whereas gram-negatives have such little peptidoglycan, we say they have no cell wall, but instead they have an outer membrane, and that contains lipopolysaccharides and porins, and they have a large periplasmic space. Now there's some meaning to that, and I alluded to those too. Why do we need to know this? Well, we don't just gram stain it to know if it has peptidoglycan. Remember, peptidoglycan can be recognized by our immune system, phagocytes, white blood cells, that engulf and destroy foreign material. And because peptidoglycan is unique to bacteria, it can be recognized by them. Also, peptidoglycan is a very good target for antibiotics because only bacteria produce peptidoglycan. Next, on the flip side, gram-negatives, they have an outer membrane that is selectively permeable. Well, if it is selectively permeable, meaning only water and uncharged molecules can pass through, that also means that they can somewhat regulate whether or not antibiotics make it through. Now, I mentioned the things hanging out of the cells. I mentioned the teichoic and lipoteichoic acid of the gram-positive cells. Those are what we call an antigen. An antigen is anything that stimulates an immune response. And so, in gram-positive cells, not only do they have peptidoglycan, but also that teichoic and lipoteichoic acids that stick out there, those acidic sugars, are immediately recognized by the body. Things like our phagocytes, our white blood cells, and inflammatory compounds. Where on the flip side, gram-negative cells have those lipopolysaccharides, the lipids stuck down in the outer membrane, and these polysaccharide side chains, these long chain sugars that stick out of there. I will highlight them here. these little tails here, those guys are what we call an endotoxin. They are only toxic when they are released by the cell. They are only toxic when the cell lyses or explodes. So they are not toxic when they are within the cell envelope. Endo meaning within. So they are a toxin within the cell envelope, and they're only toxic when they are released. Now, what happens when they are released? If you were to have a large gram-negative infection and you treated that with a broad-spectrum antibiotic that killed them all at once and released all of that endotoxin at once, that could result in a systemic effect of your patient, which means that your patient could have changes in heart rate, blood pressure, fever, diarrhea, vomiting. It's a systemic shock specific to this endotoxin. It's called endotoxic shock. So now when you look at a gram stain, what does it mean to you? What does that tell you? Does this organism have teichoic acid or lipopolysaccharides? Teichoic acid, yes. And would this organism be immediately recognized by the immune system? Yes, because of the peptidoglycan and the lipopolysaccharides. Would the patient infected with this organism be at risk of endotoxic shock? No, because it does not have lipopolysaccharides. It's gram-positive. Okay, there's always a weirdo, right? And in this case, there's a few of them. I mentioned that gram stain divides almost all bacteria into two groups, gram-positive and gram-negative. But of course, there have to be a few weirdos. Starting with acid-fast bacteria. Acid-fast bacteria are technically sort of gram-positive because they have a thick layer of peptidoglycan and no outer membrane, but instead they have a very thick layer of what's called mycolic acid. Mycolic acid on the outside of that peptidoglycan. And that's a lipid. That mycolic acid is a waxy lipid. And these guys are significant to us because they are, some of the species that are acid-fast are the organisms that cause tuberculosis and leprosy. Mycobacterium tuberculosis. Mycobacterium is a genus. The species is tuberculosis. And then some Nocardia species in the genus Nocardia can cause leprosy. Now, because they have that unique lipid, mycolic acid, and it's waxy, it contributes to their virulence. It is very difficult for the body to recognize them as being foreign because wax is very inert. It's also very difficult for phagocytes to engulf and degrade that waxy mycolic acid, again, because wax is inert. It also makes it very difficult to stain them. We can't stain them with a typical gram stain. If you were to take a candle, which is waxy, and dip it into stain, you would end up with the same color candle you started with because the stain would just bead up and run off. And that's what happens with these cells, which means they're very difficult to stain and difficult to diagnose then as well because we can't see them with a typical gram stain. So instead we use what's called an acid-fast stain. An acid-fast stain is another differential stain, but in this case we apply the primary stain, carbofuran. At the same time, we use heat as our mordant. So no longer iodine and crystal violet. Instead, we use carbofuran and heat. And the heat is going to soften that waxy outer layer and really drive the stain into the cells. Now, because all cells, non-acid-fast bacteria, acid-fast bacteria, human cells, all cells in our sample have been subjected to this application of heat, we need to use a stronger decolorizer. So instead of using just alcohol like we do with the gram stain, we use acid alcohol as our decolorizer. It actually includes 5 to 10% hydrochloric acid with the alcohol. And so that's where these guys get their name, because the a combination of acid and alcohol will strip the carbofusion out of all the cells except the acid-fast bacteria, the ones with the waxy layer. Those guys hold fast to the stain in the presence of alcohol, or acid, excuse me. They hold fast to the stain in the presence of acid. That's where they get their name, acid-fast. They hold tight to the stain or fast to the stain in the presence of the acid alcohol. And then again, now because we have a differential stain and we've completely stripped the carbofusion out of the non-acid fast bacteria, we have to apply a counter stain. In this case, we apply one of the weakest stains that we have in our repertoire, and that's methylene blue. That will stain all of our non-acid fast cells, either non-acid fast bacteria or human cells. And so you can imagine this is another sputum stain. This person has tuberculosis, and you can see the bright red Mycobacterium tuberculosis popping out there. But if we had done a gram stain on this, those cells would not stain. They would have been colorless. And the human cells, instead of blue, would have been light pink. So we would have been searching for colorless bacteria in a sea of light pink. It would have been next to impossible to spot them. So this differential stain, the acid-fast stain, lets us see those organisms much easier. More weirdos. The other prokaryote. Oh, I see, I miss an R. The other cell wall weirdos, archaea. So remember I said positive purple peptidoglycan prokaryotes. Well, the truth is, these guys mess up my alliteration because archaea do not have peptidoglycan. Only bacteria have peptidoglycan. And remember, archaea are our extremophiles. They are a very, very diverse group. So we really can't say anything else that covers all of them, that describes all archaea in one statement, other than they do not have peptidoglycan. Because sometimes their cell walls are made of protein or polysaccharides. Some of them have no cell walls at all. You have to remember that because they're extremophiles, they may have to live in wildly different conditions, really hot, really cold, really acidic, really basic, crazy pressure, crazy toxicity, and so their cell walls have to be different to adapt to those extreme climates. Now, one more, and students always mix these up, which is why I put archaea in the middle there. We're back to bacteria. These are mycoplasmas. This is a genus of bacteria, mycoplasmas, and I want you to pay attention to plasmas. Plasmas. These are different from mycobacterium. These are mycoplasmas. These organisms have no cell wall whatsoever. They are just a plasma membrane or a cell membrane, just a cell membrane or a plasma membrane. If you have no cell wall, you have no defined shape, you are pleomorphic, many shapes. And so what do they do in order to make their cell membrane strong enough so that they don't explode? They put sterols. Sterols are more lipids. You may know cholesterol. Something that kind of clogs up our arteries because it is a relatively sticky inert lipid. So again, we have cell membranes with sterols stuck in it to make it resistant to lysis. But also one of the things that they do is they just don't grow very big. If you're not very big, you're at less risk of exploding. So they're known for being very small and pleomorphic. Now these guys are important to us too because if you have no cell envelope or no cell wall, you have no real target for antibiotics. Remember, a cell membrane is present in all cells, including eukaryotic cells, our own cells. So if we were to target these guys with a drug that targeted their cell membrane, we would also be taking something that would be toxic to our own cell membrane. And they're also difficult for the body to recognize. Our type species, our example of this is Mycoplasma pneumoniae, which causes atypical pneumonia or walking pneumonia. It's hard for the body to recognize, it's hard to diagnose, and it's hard to treat, which is why it's called walking pneumonia, because people seem to have a chronic, persistent infection of this particular organism. Also, if they're extremely small, they can be inhaled extremely deeply and get seated in that lung much more easily. Okay, back to the cell envelope. So when I talked about the cell wall and the periplasmic space, and I talked about some weirdos, mentioned these things. Now, when we talk about the cell envelope, we have to talk about the cell membrane. All cells have a cell membrane, and all cell membranes, all membranes, are composed of phospholipid bilayer. They can also be called cytoplasmic membrane or plasma membrane, as I have mentioned already, composed of a phospholipid bilayer with proteins and some polysaccharides stuck in there, and they have what's called a fluid mosaic model. Now, a fluid mosaic model, I don't know how else to describe this except to ask you to imagine a kid's swimming pool. A kid's swimming pool filled with kids' toys. So you've got noodles, and you've got rings, and you've got the floating ducks, and balls, and beach balls, and everything in there. And all of that stuff floats. It's all floating on top of the swimming pool. And the kid can jump in and make a huge cannonball splash, and everything just keeps floating on top. But it's fluid mosaic, meaning the pool noodle may start out over by the ladder at the start of the day, and it may work its way over to the pool filter by afternoon, and the rubber ducky ring toy may work its way from the sunny part of the pool over and through to the shaded part and throughout the day. And so the same goes with these proteins. These proteins can start out in one part of the cell membrane and work their way to other parts. That's a fluid mosaic model. So what does the cell membrane do? Well, it regulates transports of molecules. Remember, water and uncharged molecules can pass through a membrane, so it is semi-permeable or selectively permeable. So it can regulate what comes and goes. But then also, let me ask you, what is the powerhouse of the cell for our cells, eukaryotic cells? What makes ATP? The mitochondria make ATP. Now remember, prokaryotes, bacteria, do not have mitochondria. They don't have membrane bound organelles. We're going to get into that in the next topic. So, where do you suppose they make ATP? They have to make ATP, right? Well, I bet you guessed it, they do it in the cell membrane. So this is the site for a lot of metabolic activity, especially the electron transport chain, which is where most of our ATP is made. So the electron transport chain is located in the cell membrane in prokaryotic cells. Now here's some other things that are meaningful when we do a Gram stain. When we're talking about the cell membrane, alcohol can dissolve membranes, right? That's how we do a Gram stain, and we can also destroy microbes using alcohol. That's why we use rubbing alcohol to disinfect injection sites or skinned knees, because it can dissolve the lipids in the cell membrane and kill these cells, including Gram negatives. But also, if we were to target the outer membrane of a Gram negative cell, because it's composed of phospholipids, and because all cells have a cell membrane composed of phospholipids, prokaryotes and eukaryotes alike, if we were to give somebody a drug that would target the outer membrane of a Gram negative cell by targeting those phospholipids, we may also be taking a drug that's toxic to us. So more information is taken from a Gram stain other than just positive purple peptidoglycan. So let's practice really quickly. Which of these organisms would be immediately recognized by the body as being foreign? A, gram-positive. Which of these organisms has mycolic acid in its cell envelope? C, that's our acid-fast bacterium. Which of these organisms, if a person were to be infected by them, would contain an endotoxin, and the person may be at risk for endotoxic shock? B, which is our gram-negative bacterium. And which of these organisms could be destroyed by using simple rubbing alcohol? All three of them. Okay. A couple questions here for you. I didn't write these, but why are alcohol swabs used in the treatment of microbial skin infections? Because they dissolve lipids in the membrane. And if you think back to video 1, when I was talking about the two types of cell envelopes and how I was mentioning that there are two types of anchors in a flagellum, but all gram-positive and gram-negative have an anchor, a hook, and a filament made of flagellin protein. Now you can actually say which of these anchors belongs to the gram-positive cell. And that would be B, positive purple thick peptidoglycan layer. Okay, so we've made our way through the cell envelope, and we're going to move on to internal structures, and I'm going to do that in the next video.